So in this example, we're going to do a dihybrid cross in Drosophila, the common fruit fly, and we're going to have an autosomal gene and a sex-linked gene. And we're going to look at the F1 progeny, the F2 progeny, and what happens when you cross the F1 back to the parents. And so, let's just start off by reading. In Drosophila, vestigial, partially formed wings are recessive to normal long wings. Now, stop right there. Note what kind of traits we're looking at. We have wings, and we have our recessive form, which is vestigial, and we have long wings, which are the dominant. And so let's give it VG as the way to describe this. VG, recessive, and when we're going to describe the long wings, we're not going to do a capital V, we're just going to go VG plus. Now, let's keep reading. And the gene for this trait is autosomal. So we know that this works, it just behaves properly. We have two alleles of this form, and both males and females. Now, the gene for the white eye trait is on the X chromosome. Now that we know that, we have our eyes, right? And we have white eye, so right here, is on the X chromosome. So let's just depict this with W. This is the recessive form, right? And this is a homozygous white-eyed long-winged female. It's crossed with a homozygous red-eyed vestigial winged female. So red eye is the other trait, and this is when it's dominant. We're not going to show two traits because as long as you have the dominant allele, it's all good. Now, one thing to note with this X-linked gene, if you have a male with only one trait, with one allele, since he has a Y chromosome, it only takes one of those alleles to determine his trait, right? So, when we're going to write the genotypes of the parents, we have to consider the fact that males only have one X chromosome, and so we have a homozygous white-eyed long-winged female. So homozygous white-eyed, and so we have WW, so we have two X genes, two X chromosomes, and a female, and long-winged. He's homozygous for both, and so we have VG plus, right? And it's crossed with a homozygous red-eyed vestigial wing. Now, if you're homozygous red-eyed on a male, you actually only have one allele for the dominant trait, the red eyes, because you have a Y chromosome. This is how we depict our Y chromosome. And we have vestigial wing. So it's just VG, without the plus. And we want to see what the genotype of the F1 flies are. And so let's start considering the genotype of the F1 flies. If we cross these two, we know that they're on different chromosomes. So we can just do Punnett squares for each one independently, and we just multiply the probabilities through to find what the genotypes of the F1 offspring are. And so for our sex-linked gene, our eyes, we can just do a Punnett square for that, and we get that we have heterozygous females that are red-eyed, right? So we have 1/2 red-eyed females. And then we have over here all recessive males. And so we have 1/2 white, and they're males, right? Now for the wings over here, we have VG plus, and then another VG plus. This is for long wings. And then we have our vestigial wing male. When we cross these out, we get the same result all over. So we have VG plus VG, VG plus VG, and so on and so forth. So our probability of getting a heterozygous long-winged, whatever male, female or male, long-winged fly essentially, is 1. It's 100%. It's going to happen all the time. So our progeny, our F1 progeny is going to look like this. We can do a branch diagram where we have, let's start off with doing the wings. We know that for 100%, they're going to be long-winged, right? Which is basically our heterozygous genotype over here. Not homozygous dominant, not homozygous recessive, or heterozygous. And then this can give us two possibilities. Either they're 1/2 red and female, or they're 1/2 white and male. They can't be both at the same time. And so you multiply these probabilities out, and you get that 1/2 of your progeny is long-winged, red, and female. And the other progeny is 1/2 long-winged, white, and male. Now that's the F1 progeny. Now what happens when you get the F2 progeny? How do you get that? Well, the F2 progeny is when you mix the F1 progeny together. And so we basically have a long-winged red female crossed with a long-winged white male. And we know that the genotype of the long-winged red female is heterozygous for the I gene and heterozygous for the wing gene. And this is going to be crossed with our male, which is white. We have only one allele for the eyes and a Y chromosome, and he's also heterozygous for our wings. And so now we have our Punnett square for the eyes trait. And when we mix it together, we get a heterozygous female, we get a homozygous recessive female, we get a dominant male, and a recessive male. And so 1/4 of this progeny, essentially, or the F2 progeny, 1/4 of it is going to be a red female. The other 1/4 is going to be a white female. The other 1/4 is going to be a red male. And then so on. We have, last thing is 1/4 white male. That kind of sounds weird, but yes, 1/4 white male. Now, for the wing trait, we have 2 heterozygotes, or that's a heterozygote right there, being crossed together. And when those are crossed together, we're going to get 3 different genotypes. We're going to get a homozygous dominant, a homozygous dominant over here, 2 possibilities for a heterozygote, and then we have a homozygous recessive. And so we have 1/4 of them are homozygous dominant, so I'm just going to say long-winged. Or let's go by phenotypes. We have 3 different genotypes that give us a dominant phenotype. And so we're going to say 3/4 long-winged, and we're going to say 1/4 short-winged, or vestigial wing. So we'll go vestigial. And so these are our phenotypes, and our genotypes are shown here. Now in the genotypes, we have a 1:2:1 ratio over here. But when we're going to do our branch diagram, we just want to look at the phenotypes in this case scenario. And so we're going to start branching it. We know that we have 2 possibilities. First, we know that we're either male or female. So it's 1/2 male or 1/2 female. And then we know that the male is either red or white. And notice that I already multiplied these through earlier on when I was doing this Punnett square. See how I said 1/4 red male and 1/4 white male? It's the same thing if I multiply the male with red, and the same thing if I multiply male with white. You get 1/4 for either one. But here I'm going to show you like from the scratch. So female, 1/2, it's either red or white. And then for those, we can branch it out. And so for a male who's red as well, he has a 3/4 chance of being long, and then 1/4 chance of being vestigial. And that goes for the same for all those other possibilities. It's either 3/4 long, 1/4 vestigial, so on and so forth, right? And so our final progeny for F2, we get 1/2 male, 1/2 red, and 3/4 long. And so we get 3 out of 16 is going to be male, red, and long wing. And the other, we're going to have a 1. We have 1/2 male, 1/2 red, 1/4 vestigial. So 1/16 is male, red, and vestigial. And it's going to be the same probabilities for white as well. So we already said male, right? They're going to be white now. They're also going to, and it's going to be the same probabilities for long and vestigial. So long and vestigial. And notice these probabilities are going to add up to 8/16, which is actually 1/2. And that's because our female side over here is going to be the exact same probability list. It's going to be 3/16, 1/16, 3/16, and 1/16, right? And it's just going to be female in this case scenario. And that's going to go down. And it's going to be the same list: red long, red vestigial, white long, white vestigial. And so instead of writing these out, I'm just going to do this to depict it. But I would suggest you write it out if you really need to look at it all. But all these probabilities are going to add up to 1. And so that's basically the 8 different possibilities that we can have for our F2 progeny. Now notice that there's actually more genotypes than just 8, because for our males and females who have long wings, we have to notice that the long wing phenotype is actually depicted by two different genotypes. We have 1/3 of the genotype is homozygous dominant, and 2/3 of it is heterozygous. Now, the final part of the question is asking, what happens if we take our F1 progeny and cross it back to our parents? Now, what was the F1 progeny? Well, it was just heterozygotes at both traits, and our parents were just homozygous at both. And so our female, let's do the first one. When we have our female parent, she was homozygous recessive white, right? But long-winged. And our male, in our F1 progeny, was just heterozygote. He was 1/2 white. He was also long-winged. And so we have our traits right here. So we have our female from F1 parent. So this was our parent right here. And if we cross it with its child, which was a white male who was heterozygote for our vestigial and long-winged trait, we get this Punnett. We can do the same thing. It's a Punnett square. We have WW here, WW here, and then we have little w and a Y chromosome. And we notice that all our progeny are going to be white-eyed. And so, whether you're male or female, 100% is going to be white-eyed. And we know that it's going to be half male, half female, right? Now, with our wings, we have our homozygous dominant over here and our heterozygote. And we notice that if we do this cross, all of them are going to be dominant. And so, writing this out, we have half of them are going to be heterozygote, and the other half is going to be homozygous dominant. And so we have 100% is going to be long-winged. And so for the cross between the parent and the F1 progeny, when we're doing the female parent and the male F1, we get 1/2 is going to be male, white, and long-winged, and the other half is going to be female, and then same thing, white and long-winged. Now, what happens if we do the other cross, where we had the male parent, right, who was red-eyed and vestigial-winged, with our F1 female, who is actually, if we double-check, was a red female. It was heterozygote, and was also heterozygote for our long-winged trait. If we do this cross, then we have over here our female, and then we have our male. And we notice that we have all the females are red-eyed, and then half the males are red-eyed, and the other half of the males are actually white-eyed. And so we have 1/2 of them are going to be female and red-eyed, no matter what. And then we're going to have 1/4 is male, red, and the other 1/4 is male and white. If we do the cross for our wings, we have a heterozygote and a homozygous recessive. We're going to get half of them is going to be our dominant trait, and the other half is going to be our recessive trait, which is vestigial wings. So we're going to have 1/2 long and 1/2 short. And so now if we multiply these out, essentially if I do a branch diagram, your final results are going to be 1/2 female red times 1/2 long. So we're going to have 1/4 female red and then long. You're going to have another 1/4 is going to be female, also red, but short, or vestigial winged, I guess. We'll go vestigial. And then we have 1/4 male red times 1/2 long. So we're going to have 1/8 male, red, and then long. The other eighth is going to be male, so same thing. Red also, but short, so let's go vestigial. The other eighth, also male, but now it's going to be white. And then same thing, long or short, and the other eighth is going to be male, white again, and we're going to have short. And so that's going to be the genotypes and the phenotypes of the F1 crossed back to its parents. And so we have two different results. You can have either the result when you cross the mother parent with the F1 male or the father parent with the female. Okay. And so this video was long, but these are all the different examples that you can have on a typical dihybrid cross.